Hello everyone, and welcome to today's live broadcast. A neovirus lifestyle exhibited by a positive ssRNA virus hosted in an unrelated dsRNA virus. Taxonomic and evolutionary considerations presented by Nobuhiro SUZUKI, Professor, Institute of Plant Science and Resources at Okayama University. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you are having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Nobuhiro SUZUKI. I will now turn the presentation over to him.
Thank you, Marge, for your good introduction of myself. My name is again Nobuhiro SUZUKI from the Institute of Plant Science and Resources, Okayama University. My talk is composed of two parts. The first one is introduction of fungal viruses, particularly infecting filamentous fungi to you. And in the second part, that is the main part of my talk or presentation, I try to convince you of a new virus lifestyle or unique virus-virus interactions or interplays in the white root rot fungus, which is plant pathogenic fungus. So first, this slide shows you where I'm from or where I am now. So I'm based in Kurashiki city in Okayama Prefecture, which is located west, roughly 200 kilometers west of Kyoto. The distance is just 1 hour ride by bullet train from Kyoto. So Kurashiki city is famous for its historical view shown here. The city tried to preserve old-fashioned buildings built in Edo or Meiji eras. And this city is famous for denim or jeans. So our institute shown here is in the downtown of the city. I'm now in this building shown here. But this institute is over 100 years old. Okay, back to science. So first, let me introduce fungal viruses. I'm sure most people or most audience is unfamiliar with viruses infecting fungi. So it's believed that over 1 million species, fungal species in the globe. And not all fungi shown here are tested for virus infection, but some were tested previously, and they show that a few percent to over 90% of field isolates of each fungal species tested are virus infected. In other words, each fungal species has their own virus world with great diversity. Among 1.5 million fungal species, only a limited number of fungi are pathogenic to human or plants. So far, about 50 fungal species are known to be human pathogenic, and roughly 10,000 fungal species are plant pathogenic, exemplified rice blast, or chestnut blight, or powdery mildew fungus, shown here. You may wonder why we study fungal biases, very minor area of virology. So my laboratory has been working on fungal biases for many years for a few reasons. Shown here, these are not all, but some. So fungal bias studies can contribute to understand bias diversity. In fact, the past decade, we have shown that there are new virus families in fungi. Examples include Megaburner-Viridae, Botiburner-Viridae, and Cordyviridae. So such studies lead to discoveries of new virus structures, particle structures, and gene expression strategies. Fungal virus studies also contribute to understanding of viral replication and symptom expression, and have positive impacts on other virology, like eukaryotic virology or plant virology. For example, fungi have, like other eukaryotes, antibiotic defense responses. So far, two defense mechanisms are known. One is RNAi, RNA silencing, which is conserved across eukaryotic organisms. The other one is vegetative incompatibility, which works at the population level. So studies for such topics can stimulate the similar areas of other virologies, like plant virology or eukaryotic animal virology. And lastly, if fungi are pathogenic to plants or crops or cattle or animals, viruses infecting them may be able to be used as biological control agents, as demonstrated in hypoviruses, which can successfully control chestnut blight fungus in Europe and US. And other examples include white root rot, a biocontrol, which is testing in Japan, and also Sclerotinia sclerotium in China. These are still experimental, but promising approach for control of plant fungal disease. So let me remind you of general features of fungal viruses. Fungal viruses are different from plant or animal viruses. This slide summarizes the difference and general properties of them. Fungal viruses have no extracellular route for entry, so very different from other viruses. And their transmission is limited to vertical via spores and horizontal via hyperfusion. So let me explain a little bit more about horizontal transmission because I touch this at the later stage of my talk. So as you know, filamentous fungus can make network by fusing one mycelium to another, as shown here, which is called anastomosis or hyperfusion. By doing this, cytoplasmic material of one hyphae can move from one hyphae to another. That include viruses and organella. But this hyperfusion occurs only between vegetatively compatible mycelia or colonies. So if the two colonies are compatible, as shown here, virus shown by V+, can be moved from one infected colony to originally uninfected colonies, shown here. So this side was originally virus-free, shown by V-. So this virus transmission occurs in compatible combinations. However, if two colonies are vegetatively incompatible within one single species, virus can't move from one side to another because after the hypercontact, programmed cell death occurs, which hampers virus transmission, shown here. So the phenotype of two colonies stay the same without virus transmission. Okay? So back to general features of fungal viruses. They show persistent infection. So fungal viruses persist eternally, basically. And most fungal viruses have DS double-stranded RNA genomes. Okay? And some of fungal viruses form rigid particles, but others not. So this point is very different from other usual viruses infecting animal or plants. And cell-to-cell movement is possible via septal spores, which connect one cell to another. Through septal spores, even nuclei can move. So in the second part, this is the main topic of today's presentation. I try to convince you of a new virus lifestyle shown by two fungal viruses. Okay? Viruses can be defined as shown here, probably a very simple way. Viruses are infectious nanoparticles encasing genomic nucleic acids. However, the past 2 decades, huge or great discoveries occur which challenge these virus rules and concepts. One of them is capsidless viruses, which are unable to form rigid particles, as I mentioned for fungal viruses in my previous slide. The second one is giant DNA viruses. I can show you these exceptional viruses. Okay, capsid-less viruses are mostly found in fungi, exemplified hypovirus and nanovirus and endonaviruses. They don't make typical virus particles like spherical particles, but instead their genomes are encased in host-derived vesicles, shown here. This is proposed for hypoviruses. The second viruses that challenge virus roots are giant DNA viruses. These viruses exceed some bacteria in the size of the genome and particles. This virus has over, as you see here, 0.5 micrometer in size, and their genome is over 1.2 megabase pair. And their examples are shown here, like Pandora virus and phytovirus. So these are examples challenging the virus roots. The past 5 years, we had also a chance to discover such virus, which shows new virus lifestyle, which is a capsid-less, positive sense, single-stranded RNA virus hosted by an unrelated double-stranded RNA virus. So this relation is complex, but this slide illustrates the relationship of the two main viral players, Yado-Nushi virus and Yado-Kari virus. If you are Japanese, you can readily know the relationship, or you can guess. But for those of you who are non-Japanese, Yado-Nushi literally means in Japanese landlord or room owner. Okay? While Yado-Kari means borrowing a room to stay, or hermit crab biologically. So you may be able to guess now the relationship between the two viruses. So let me explain the new lifestyle of these viruses. So where these viruses are found is one of the plant pathogenic fungi, Rosalinia negatrix, which is notorious because this fungus is destructive and spreading in the soil in the form of mycelia. And this fungus kills the host tree. This is the example, one of the examples of diseased tree, cherry tree, almost dying. And sometimes this fungus destructs the entire orchard. And this fungus has a wide host range, over 400 plant species. To control this disease, farmers pour chemicals, shown here, 50 to 200 liters per tree. So it's very time and labor costing. So in the 1990s, Dr. Matsumoto's team proposed viral control. It's one form of biocontrol using viruses infecting pathogenic fungus. So he and his team collected over 1,000 field isolates of this fungus from all over Japan to find virus with biocontrol potential, which means the virus should be able to reduce virulence of the host fungus to use as a biological control. So the results are shown here. They found that 20% of collected fungi have viruses, singly, doubly, or triply, which means co-infection with more than one virus. So we joined this project in 2009, and we are given one of such virus-infected fungal strains, shown in this slide. So the fungal strains 1032, shown here, this field isolate is reduced in growth rate, shown here. And also this is hypovirulent. In other words, this fungus lost the ability to weaken the host apple tree. I can show you later. And importantly, this fungus has a few dsRNA bands shown here, which could be the genome of viruses. So our laboratory took a closer look at this fungal strain, 1032. First, we purified virions from that strain, and we tested it using electron microscope. As shown here, the purified fraction has one type of particles, which is 40 nanometer in diameter, spherical particles. And this purified fraction has 3 dsRNAs, shown here. And this fraction has one single protein band when examined by SD's Page protein gel. And then we sequenced these 3 dsRNA elements by using conventional dsRNA sequencing method. So the largest one was turned out to be the genome of a dsRNA virus. We named it Yadonushi virus 1. The genome size was almost 9 kb, with 2 open reading frames. The second 3 proximal open reading frame, 2, encodes RNA-dependent RNA polymerase, which is the hallmark of RNA viruses, so responsible for RNA synthesis, replicase. The second one, the very faint band, was shown to be a defective RNA of Yadonushi virus, the first one. So this dsRNA lacks RDRP domain, thus being defective, which is unable to replicate without helper virus, Yadonushi virus. The third one, the thick band, was shown to be the genome of another virus, Yadokari virus, which has positive sense single-stranded RNA genome. The size is 6.3 kb, shown here. But please remember, this virus fraction has one type of virus and one type, probably one protein band. And the phylogeny of two viruses are shown here. The first virus, ssRNA virus, Nuss virus, clustered with other ssRNA viruses shown here. Whereas Yadukai virus shows phylogenetic affinity to calicic viruses that include notorious Sapo noroviruses, pathogenic to human, causing diarrhea sometimes. So two viruses, Nuss and Yadukai viruses, are totally different phylogenetically. So back to the protein component of 1032 virus fraction. As I mentioned earlier, this fraction has only one single band. We analyzed this protein band by two approaches. One is peptide mass fingerprinting, and the other one is N-terminal sequencing. So this protein was mapped to 3' portion of the open reading frame 1 of the Nuss virus, shown here. And starting at this place. But importantly, no peptide fragments were mapped to other open reading frame, including Yadukai virus. So if you are a virologist, you may predict this Nuss virus CP trans-encapsulate Yadukai virus. So we tested this hypothesis using immunoprecipitation. First, we prepared antibody against recombinant Nuss virus CP and used that for immunoprecipitation of 1032 dillium fractions. So please look at here. The input is shown here, 1032 dillium. So anti-Nuss virus CP can precipitate some fractions, portion of virions, and that precipitates contain Yadukai virus RNA and of course Nuss virus RNA, dsRNA shown here. And also Nuss virus CP antibody, of course, can penetrate Nuss virus CP as expected. But important point here is Yadukai virus RNA is also immunoprecipitated. So this result indicates that Nuss virus CP hetero-encapsulate Yadukai virus RNA. Actually, not only Yadukai virus RNA, but also Yadukai virus RDRP encoded here is encased by Nuss virus CP, which was tested by Western blot analysis using Yadukai virus RDRP antibody. For this experiment, we prepared two virion fractions. One is original 1032 virion fraction. The other one is isogenic fungal strains 563 infected with Nuss virus 1 alone. I'd explain this, how we obtained this viral strain or fungal strains later. So anyway, these virion fractions were subjected to Western, and the results show here. Only doubly infected 1032 provided two bands. One is 100 kD A. The second one is smaller band, 40 kDa band. So this result showed that RDRP encoded by Yadokarri 1 is also encapsulated in trans by Yadonushi virus CP. So you may wonder whether Yadokarri virus is dependent on Yadonushi virus. In other words, Yadokarri virus relies its viability on Yadonushi virus. To test this, we took a transfection protocol shown here. This is how we artificially introduce or inoculate fungal host. First, we prepare single-cell protoplast from virus-free mycelia, shown here, by degrading cell wall. And in the presence of PEG, polyethylene glycol, and calcium ions, virions are mixed with not only virions, also we can introduce RNA or DNA using the same method. Virions are introduced into spheroplasts. At this stage, not all protoplasts are infected with the virus, but during regeneration of mycelia, the virus can systemically infect the entire colony. This is how we inoculate fungal host using protoplasts. So the results we got is shown here. We analyzed many transfectants. Some of them are shown in this gel, but to summarize the results, the transfectants were grouped into 4 types shown here. The first group carries only Yadonushi virus. Second one has Yadonushi and Yadonushi defective RNA. Third one is Yadonushi and Yadokarri. The fourth one is the same as original 1032 fungal species, having 3 dsRNAs. Yadonushi and Yadonushi defective and Yadokarri virus. But importantly, no Yadokarri virus single infection was observed. So this means Yadokarri virus probably depends its viability on Yadonushi virus. To further confirm this conclusion, we took a full-length cDNA approach. We prepared the full-length Yadokarri virus 1 cDNA, flanked by 2 ribozymes, hammerhead type and hepatitis delta virus ribozyme. The idea here is in transformants with this construct, authentic Yadokarri virus RNA are made in transformed cells. So if Yadokarri virus is dependent on Yadonushi virus, only in the presence of Yadonushi virus infection, Yadokarri virus can replicate. To test this, we took a co-culture method on PDA synthetic plates. So we let 2 fungal colonies fuse in one single plate. One side is inoculated with Yadokarri virus cDNA transformant. The second colony is inoculated here with Yadonushi virus single infectant. As I showed you before, we got, after transfection of virions, single transfectants, one of the 4 types. So if they fuse and exchange cytoplasmic materials, Yadokari virus may be able to replicate after receiving Yadonushi virus. So the results are shown here. So this side shows before co-culture, and the right-hand side shows after co-culture. So K and N denotes transformants with Yadokari and infectant with Yadonushi alone. So before co-culture, Yadokari virus cDNA transformants can produce transcripts which are produced in nuclei as expected and detectable by RT-PCR, but no dsRNA, which is replicative form of Yadokari virus. While Yadonushi virus transformant can produce dsRNA, but of course not Yadokari virus transcript. And after co-culturing, very interestingly, Yadokari virus now can be able to replicate their dsRNA shown here, together with Yadonushi virus replication shown here. So only in the presence of Yadonushi virus, Yadokari virus can replicate. So these results clearly indicate that Yadokari virus relies on Yadonushi virus for viability. Okay, but still we don't know yet whether Yadokari virus use Yadokari virus RNA-dependent RNA polymerase or replicase, or Yadonushi virus replicates. So to test this, we took the advantage of reverse genetics because we now have cDNA infectious clone of Yadokari virus. So we mutated Yadokari virus cDNA motif GDD, shown here. GDD motif is the catalytic motif responsible for RNA synthesis. And we made other GDD motif by substitution. Oh, I forgot to mention, M3 mutant has a deletion. So now GDD become GD. And second GDD mutant, GDD was changed to GAD. And we also made mutants in 3' UTR. Okay, we used these full-length constructs for transformation, and we did the same way as the previous slide. So the result is shown here. We checked replication competency, and GDD mutants were no longer able to replicate, while 3' UTR mutants can replicate. So these results shows that RDRP encoded by Yadokari virus is trans-encapsulated by Yadonushi virus CP and is functional in Yadokari virus RNA synthesis from this result. You may wonder what or whether Yadokari virus has impact on Yadonushi virus. The answer is yes. We compared Nuss virus content or accumulation in the presence or absence of virus. So please compare Nuss virus, the top dsRNA band here in the presence of virus shown here, or absence of virus. This gel is designed to compare or quantitatively compare against ribosomal RNA. So this RNA is total RNA from infected fungal host. So please compare the intensity of Nuss virus. It's evident that Nuss virus accumulation is enhanced in the presence of virus. So virus benefits the replication of Nuss virus. So the relationship between the two viruses are mutualistic, not commensal. So we have proposed the replication cycle, a model for replication cycle of the two viruses based on the results I explained to you. So Nuss virus is an independent dsRNA virus. So like other dsRNA viruses, Nuss virus encapsulate their own RNA, positive sense, and within the particle, the RNA is replicated, and double-stranded RNA encasing capsid can now serve as transcription place to produce messenger RNAs for translation of CP and CPRDRP. Virus, on the other hand, hijacks the CP of Nuss virus and trans-encapsulate not only RNA of virus, but also virus RDRP to replicate and to transcribe the genome. So virus behave like dsRNA virus, as if it were dsRNA virus, although its genome is positive sense RNA. It's a little bit complex though, but still we don't know yet. So here are some open questions shown here. We must demonstrate virus replicates in the Nuss virus heterocapsid. To this end, we took the advantage of virus infectious cDNA to show virus RDRP is used for the synthesis of virus RNA as shown before. But still we have performed at least two experiments. One is to know purified heterocapsids can serve as replication site. This is kind of biological approach. The second experiment is to determine Nuss virus CP overexpressors without Nuss virus infection can support virus replication. This is a genetic approach. Other open questions include virus-like viruses are found in other fungi. Just last year, an Italian group and also a Japanese group report similar sequences of Penicillium poetidis-like virus and Fusarium poae mycovirus too. These viral sequences are similar to virus. but we don't know yet whether partner viruses, if any, can support these Yadokari-like viruses, as in the case for Yadokari and Nuss virus. And we are also interested in evolution of Yadokari virus because we believe any virus won't be independent like human being. But at the moment, Nuss virus is dependent virus. So what was previous form or future form of Yadokari virus? Let me try our premature hypothesis for the evolution of Yadokari virus. I'm not Paul Gauguin, but we are also interested in where does Yadokari virus come from and what it is now and where it is going. So below this is our model based on previous research on other viruses and our results we got in our lab. So we believe ancestral Kalesi-like virus was the progenitor virus of Yadokari virus. Judged from its phylogenetic relation with Kalesi virus, and the progenitor virus might have lost the CP gene and encountered neutralistic partner with the ssRNA genome. So the present form of Yadokari is explained by myself up to here. Yadokari virus is dependent virus, depend on Nuss virus, and capsidless virus. So in the future, Yadokari virus may have two possibilities. One is as an independent capsidless positive sense single-stranded RNA. Yadokari virus may become like present extent capsidless fungal viruses that utilize host membrane as the replication site, just like hypoviruses or nanoviruses. The second possibility is Yadokari virus become full-fledged dsRNA virus after acquiring the CP gene from the partner Nuss virus-like virus. So these two possibilities are what we are thinking now. So this is the, I think the last slide to the acknowledgement. Some of you may be confused by other viruses or some viral agent with Yadokari virus. This table shows the list of similar viruses or sub-viral agent. Here is Yadokari virus. This virus has positive sense single-stranded RNA, but doesn't encode capsid protein. So the RNA is encapsulated by the partner virus, Nuss virus. So Yadokari virus now is not autonomously replicating, but this virus encodes RNA polymerase. Let me explain some of the other examples, like Umbrella virus. This is plant virus. This has also single-stranded RNA virus, but doesn't encode like Yadokari virus, no capsid protein. So encapsulation depends on the partner luteovirus. So very similar in this sense. But this virus can replicate autonomously and encode RNA polymerase. So without partner, this virus can systemically infect plant. So this point is very different from Yadokari virus. And if you are interested in animal viruses, I can show you the difference between hepatitis delta virus and Yadokari virus. Hepatitis delta virus has a circular negative sense RNA genome and doesn't encode major capsid protein, so encapsulated by hepatitis B virus. This virus doesn't encode RNA polymerase, so this virus uses host DNA-dependent RNA polymerase. So this virus is not able to replicate autonomously without hepatitis B virus. So now you see Yadokari virus is totally different from other viruses or viral agents. I hope I can convince you of the new viral lifestyle exhibited by Yadokari virus and Yadonushi virus. To wrap up my talk, I'd like to thank lab members shown here, Drs. Kondo and Rui-chan and Hisano. These two people were directly involved in the results I explained to you today. And fungal strains are provided by Satoko Kanematsu, and other collaborators are highly appreciated. Thank you so much for your patience. I hope you understand my talk.
Thank you, Dr. Nobuhiro SUZUKI, for that informative presentation. Unfortunately, it looks like we are out of time. However, if you do have questions you'd like to ask, you may click on the green Q&A button at the lower left of the presentation window. Type your questions into the box that appears on your screen and click on the send button. He will answer as many of your questions as he has time for via email. I would like to once again thank Dr. Nobuhiro SUZUKI for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.